Thank you for the nice presentation and that nice introduction that I can present my work here. I'm talking now about digital tissue deconvolution. How is this moving? Oh, it's not like reacting. Or do I have to click here always?
Sorry, laser pointer.
No, and it's like also the slides are not moving.
Sorry, just a moment.
Okay. Wait, okay.
Just right click it.
Okay. There are tissue deconvolutions. That means that we have normally bulk profiles, which are consisting of several cells, and we have a reference profile matrix where we have for different cell types references, where we have like for every cell type like the normal the expected gene expression, and what we want to predict is the cellular composition sees that we have several proportions of the immune cell populations. But why do we want to know this? There are several reasons for this. For example, that we want to resolve the source of differential gene expression, or we want to provide information for treatment decisions of medical doctors. So a naive solution of this problem would be that we just write this equation and minimize it via the cellular composition C. But in reality, this is not so easy. We have to deal with several problems, like it can be that we have immune reference profiles of two immune cell types which are very similar. This is then difficult to disentangle, or that we have a cell type which is very just a small amount in the bulk profile, or we have missing reference profiles. That means there are more immune cell types in my bulk probes than I have in my reference matrix. So we have to now first look at the notations that we can go further. We have the bulk profiles Y, where we have, this is an example for one bulk profile. In the matrix, I would have here several profiles, one after the other in the columns, and in the rows, I have the gene expression. Then I have my reference matrix X, where I have my cell types in the columns, and again, the gene expressions in the rows. And out of this, I want to calculate the cellular compositions. For every bulk profile, I want to have the cellular composition into the Q different cell types. So first we start with a formal publication of our lab. Here we said, okay, we introduce gene weights G, which are chosen in a way that I improve the estimation of my cellular compositions, so that my auto loss function, which is the correlation between the true cellular composition and the predicted cellular composition, is minimized, so the correlation between these two is maximal. So I always need training data where I know the cellular composition that I can train and learn my gene weights. But now, when we have like the bulk profiles and the reference profiles, which are not covering everything of my bulk profiles, I will have a biased cellular composition. So what we introduced are as an estimation of background contributions. And here we go to SQL work of Raclair, who introduced the program EPIC, and we use this notation. He said, okay, my bulk profile here is now the reference profile times the contributions, plus a contribution from a potential hidden background. X, small as a small x, is a reference profile, and the small c is the cellular contribution plus an error. So we can rewrite this, that we can estimate our cellular contribution from the hidden background by 1 minus the sum over the contributions of the cell types which are covered by the reference matrix. And there's an intrinsic assumption in EPIC that we said today, the cellular compositions of the covered cell types, they're independent from the background profile and background contribution. But we release this assumption and say, okay, now there can be an exchange between them. And though we insert the formula for the hidden background contribution in our loss function here and do a regularization on it. But are we now done? No, because it's also said like we know that cells are adapting to their molecular phenotype and the environment. So it means when we have immune cell types and a cancer, these immune cell types will adapt to the cancerous environment. And also when we compare two different tissue types, a T cell in a lung T cell will have a different expression profiles than a T cell in the blood or skin or something. So global or static reference profiles might be inappropriate. And our solution is that we adapt the reference profiles dynamically to the specific application. And this is that we replace our reference profile by a rescaling matrix delta, which has the same size like the reference profile matrix. And we just replace it by delta Hadamard X. And so we introduce this into our loss function learning program and do a regularization term in addition. The only thing which is important here is that you have to keep in mind the delta matrix encodes for gene regulation. So I start with a delta matrix of 1s, and then I get for us cell type I and gene J are value above 1, then it means in this cell types, the gene J is upregulated. When it's below 1, it's downregulated. This is our algorithm. We start with reference profile and bulk profiles, and then we update the cellular composition, we update the hidden background profile, and then we update the regularization matrix for every gene until we have our desired precision achieved. Then we are testing, validating this. And first we start with the training and validation set. And here we use healthy single cell data of breast tissue, and we're creating artificial bulks. In our training data, we have 6 cell types, we have no hidden background, and this we're using to learn our gene weights. And with this gene weights and the reference profiles, we go to the validation set where we have the same cell types included, and in addition fibroblasts, and calculate then the cell type distribution, the contribution of the hidden background, and the profile of the hidden background. And here we see, like, we compared our method to several other methods. This is the results of our method. This is EPIC, where we gave EPIC our reference profile. This is EPIC, where we used the reference profiles EPIC was providing. This is Cybers.X and Skaten. And we can see that here are the deconvolution correlations between true and predicted cellular composition for the cell types in the reference matrix. And we can see that we get good results for all cell types, and we're outperforming in the mean all other deconvolution methods. But we talked there was a hidden background, and though, what is with the hidden profiles? Here we see the correlation for the contribution of the hidden background for ADTD and EPIC, because these are only 2 methods which are providing this. And we see that we're outperforming EPIC. And also for the hidden background profile, which is just ADTD is providing. And here we also get a very good correlation value. So we're able to recover the contribution and profile for fibroblasts, which were the hidden ones. Then we start the domain transfer to breast cancer. Here we now using healthy single cell data from the breast tissue like before for creating the reference profile and the gene weights and the hyperparameters lambda1 and lambda2 from the validation study. And then we now go to the test data, which is again single cell data, and create artificial bugs, which contains the same cell types like in our reference matrix from X from the healthy ones, and also add cancer epithelial cells that we have hidden background. And what we want to find out is we want to see how our method is comparing to other state-of-the-art methods, how stable is the method when depending on the sample size, like how many bulk profiles I'm using in my Y matrix, and if we can reconstruct cellular regulations. And first we look at the performance. We compare to the same state-of-the-art methods, and we again outperform all the other methods. We have good results for all cell types, except the epithelial cells, but there is a biological reason for this. In breast cancer, epithelial cells are turning to cancer epithelial cells, and so it's not always clear. This is a continuous transition, so it's not always clear if a cell is totally healthy or totally cancerous. There are also cells in between, so it's hard to distinguish. And we're regarding also the hidden profiles, and here we get also quite okay results. We are outperforming EPIC, and so we can also say we can recover the contribution and the profile of the cancer epithelial cells. Then we're looking at the sample size stability. We're now performing ADTD for different numbers of bulk profiles. This is the number of bulk profiles on the x-axis, so from 10 to 1,000, and on the y-axis we show the correlation for the 6 cell types in our reference matrix, and we get stable predictions even for very small sample sizes. We get deconvolution results for the most cell types, and but here we can see that this continuous transition from healthy to cancerous epithelial cells is yielding as a not perfect correlation performance, deconvolution performance. Then we regard the hidden background. Here we have shown the correlation for the contribution of the hidden background, and in red the correlation for the hidden background profile compared to the true hidden background profile. And here we see that we need more samples, like around 350, to have like a stable and good result for the deconvolution. And we have a good estimation of contribution and profile of cancer epithelial cells. We want also to recover cell type specific gene regulation. That means we have modify artificially gene expression levels of gene J and mixture I for a randomly drawn cell type. And we draw their modification factors and add this to our original expression, and we modify in total 60 genes, 30 very unspecific and 30 very specific. And then we see if we can detect the modified genes in our regulation matrix delta. And this we can see for different regularization parameters. We get the area under the curve, like how much of our manipulated genes are we detecting, and we're getting, especially for small values, very good results that we're detecting like 70%, 75% of our manipulations. The next step is we go now to real bulk data and not using simulated bulk data from single cells. And for this, we're using breast cancer tissue from TCGA, and we do a subtype analysis. We have triple negative, HER2 positive, luminal A and luminal B breast cancer subtypes. And what we're doing is we performing ADTD on all of these 4 subtypes and identify the regulated genes with our delta matrix. So we see what are the differences between a healthy T cell, for example, and a cancerous T cell, for example, or B cell. And here in this Venn diagram, we show the overlap among the 10 most regulated genes. We see we have some genes which are regulated in all 4 subtypes. It seems to be genes that are then differentially regulated in breast cancer. And we see also that there are genes which are regulated just in 1 or 2 of the subtypes. So it seems to be something specific for this cancer subtype. These are the heat maps of 6 of the most important regulated genes, and we see that it's not a regulation over all subtypes or over all cell types. We see there's a clear difference, like here in B2M says, for example, really just the myeloid cells regulated, and only for HER2 all those T cells. In this gene, we are now giving a closer look. B2M, we see that it's highly regulated in all myeloid cells for all 4 cancer subtypes, and then also for T cells in the HER2 subtype. It is known that this is a gene which serves for antigen presentation. And we know from literature that there's higher gene expression in cancer compared to benign lesions. This is exactly what we see. And it's also in the literature that the protein expression is significantly different in the subtypes. This is exactly what we see. So to sum up, we wanted to estimate gene weights for cell type prediction. This we can do. We wanted to estimate hidden background distribution and profile. This we also can do. And we wanted to estimate cell type specific gene regulation. This we also could show that we are able to do this. And I want to thank here my collaborators, my professor at the University of Bergen, Sushma Gralshed, the Gralshed Group, and the Lawrence Group, where I'm actually employed, and my colleagues from the University Medical Center in Göttingen, especially Michael Altenbuchinger, Malte Mensching-Buhr, and Thomas Ster. So thank you for your attention.
Thank you for the talk. Do we have any questions?
Hi, yeah, fantastic talk. So have you guys thought about ever trying to do multiple hidden profiles at all? That
yes,
theoretically
we have
possible?
thought about it, and yes, we're developing this.
Yeah, I have just a basic question. When you are doing some of your benchmarks, in some case, you simulated a bulk tissue from the single cell data. Does that create some bias that make it look better than if you really have both types, bulk data and single cell from the same person? Because single cell data is very different than bulk in terms of sparseness.
I think if you really wanted to do, I think that when you have single cell data from one person and when you want to do bulk data from one person, this would get a better result, but this we don't have. Like we have a normal single cell data from several patients and from different patients, like this from TCGA, there we have bulk data. And this is like how you have it normally. The aim is that you say, I could train a model on single cell data for one specific cancer type where I'm interested in, and then I have learned G weights, and then I can go to, with my ADTD algorithm, to bulk data in there. And now I can predict a lot of bulks because the measurement of bulk is way, way cheaper than measuring for every patient the single cells.
I understand that's the goal, but I think from benchmarking the algorithms, would it be nice to have like a set of, 20 patients where you have both single cell and bulk?
Very nice, but
I don't know, it may be something for a consortium to develop.
This would be nice, but this is we unfortunately don't have. And you don't find it published. So we have now here to rely on published data.
Yeah. Last question over there.
Hi. Thank you for your talk. It's super useful work. I have two questions. One of them is your model available to retrain for different types of bulk tissue? For example,
this is the aim that you say. Our goal is always that, okay, I need in the beginning some single cells of the tissue I'm interested in. And like the cell types I'm interested in, when the different immune cell types, and then I train a model, and this then I have like my gene weights, and with this I can go to bulk data of the same tissue origin.
I see.
So I'm very specific, and this is what improves the deconvolution results, that I don't want to try to have an overall model for everything, but I say, okay, I can easily train specific models, and then I'm better to solve my specific problem.
Okay, and in that case, my second question will be, do you know how the accuracy changes as you kind of descend down the subcategories of cells? So when you can compare neurons to, for example, glial cells, but then there are subtypes of neurons and then subtypes of those neurons.
We are working on this. We are actually also trying this. What I can tell you is it's getting more difficult when you have similar cell types. And then you would need more genes, so it's getting more computationally demanding. And the problem is also what we talked with some people of the Oslo University, which are working with single cells, that label us some cells or gave us some labels, as in to say, the first layer you can trust.
Mhm.
The major setup. And when you go down the next, you can also trust. And the third, there's also like, it's not clear. It's like there's also the cell type labeling is not 100% clear. So this makes it also more difficult, because when I cannot give in correct label data in a model, it cannot learn perfect.
That makes sense. Thank you so much.
Okay.
Thank the speaker again.